All right, so let's go ahead and continue on with the second part of lecture number 10. Remember, we're talking about the innate immune system function, and a big part of the innate immune system function is how it communicates. Again, this is something that we kind of take for granted, that all the cells in our body are able to talk to one another and communicate with one another. But remember, they don't have eyes and ears. They don't have Twitter or whatever it might be. How do cells communicate with one another? Well, they do it through a series of a very complex series of ligands and receptors. So we have a signal. So we have so that signal, that initial molecule that starts this process, is called a ligand, and a ligand is going to attach to a receptor on some type of cell. And this cell has a specific receptor that is specific for this particular ligand. Ligation of this ligand with the receptor is going to lead to some type of signal response within the cell. And that response is ultimately going to cause some change in the cell. Now, usually it's genetic expression of new proteins, because remember, ultimately proteins are what does everything in our cells and in our body. So the response is almost always activation of transcription factors that are going to turn on very specific genes to make very specific proteins. Now, there are, goodness, I want to say thousands, but let's just be on the safe side and say hundreds, maybe even into the thousands of different kinds of ligands just in our immune system. So think about all the different ways. Almost think of them kind of like emojis, right? Like they're a very simple signal that lets someone know I approve, or I don't approve, or I'm angry, or I'm sad, or stop, or keep going, right? So think about all the different kind of messages that you can convey with a very simple emoji. That's kind of what a ligand is. So think about a ligand like an emoji, and then you're going to send that signal to someone else, and they're going to receive that signal in a certain way and do something based on that signal. So the way I kind of think about it, another way of thinking about this ligand receptor complex would be almost like, think about a big building like the S building at school. We have very very complex sensor systems. We have smoke detectors, right? In all the hallways and buildings and rooms and so on of the S building, we have smoke detectors in various areas, and that smoke detector is the receptor. And that receptor is going to be, when it's ligated by something very specific, a very specific type of smoke or some type of molecule that's going to attach to that receptor, it's going to set off a series of signals that eventually get to the sprinkler system and turn that sprinkler system on. And that's kind of how chemical messengers work within our body. Now, there's probably about a half dozen different categories of these ligand receptor classes that we can talk about. We're going to talk about 4 of them. The first one are chemokines, and chemokines are specific. So chemokines are ligands that tell other cells you're going the right way or you're going the wrong way. In other words, it functions in taxis. Taxis means moving. So if we want to recruit macrophages, if we want to recruit neutrophils and dendritic cells to the site of infection, the cells that are already there are going to secrete chemokines that tell other cells to come closer. Come over here because here's where the danger is. On the other hand, we have interferons. Interferons are warning signals. So when cells secrete interferons, they're telling any cell that picks up that interferon signal that there's danger in the area and to be on the lookout. We'll talk about a specific example of an interferon later. I think it's our last slide. The next one, interleukins, they're abbreviated IL. Interleukins are stimulating, they either stimulate or activate cells to do very specific things. And oh my goodness, I think at last I knew there were at least several dozen different types of interleukins. And they have a very complex chain of events that can lead to a specific result. For example, actually I just read an article the other day that is implicating interleukin 6, IL-6, in the response to coronavirus. So we all see in the news every day about how someone, some 30-year-old that was perfectly healthy was fine 1 week and they died within 2 weeks after the COVID-19 infection, and people are wondering why. And so everyone's trying to figure out what it is in these specific patients that have such an extreme effect, such an extreme response to this coronavirus. And a lot of the early research is saying that it's not necessarily the virus, it's the cytokine response of the body to this virus, specifically interleukin 6. Interleukin 6 is an inflammatory cytokine, and it can lead to a lot of damage in your kidneys, and it can almost lead to like a toxic shock syndrome like response from the body if we get this extreme cytokine response. And so there's some early research that shows that might be one manifestation of the coronavirus response. But generally speaking, we have to have these. And again, this is that complex interplay of the immune system. Why we have to have a response and we have to activate these inflammatory pathways, but if we do it too much, we die. And so again, very complex, very delicate system. So these interleukins, again, they activate cells. They're going to stimulate these cells to start doing very specific things. But if they do it too much, we can have too much inflammation, too heavy of a response, and we can damage our tissues. And if we damage our tissues too much, we can lead to organ failure and death. The last type of cytokine, this was the one that I studied the most actually in college, or in grad school, I should say, are cytokines called tumor necrosis factors. And they were originally, so they're called tumor necrosis factors because they were originally signals that made cells that had been transformed, in other words, either cells that were already tumors or cells that could become tumors, they caused them to die, which again is very important. Remember, we have to have cells dying in our body daily, right? That's a normal part of homeostasis. We want our cells to die and turn over. And so that normal process of death and turnover is called apoptosis. In other words, we have cells that are programmed to die, and they die in kind of an organized manner, and they get taken care of by our immune system in a healthy way. And the way that they get that signal is through these specific factors called tumor necrosis factors. Finally, we have one more type of receptor. It's not soluble. It's not like kind of free floating like our ligands are. Our ligands are. Adhesion molecules typically are part of the inner lining of blood vessels. So they are on the inside of blood vessels, and if they are expressed highly, they're going to grab onto cells of our immune system and make them kind of recruit them to the site of infection. So all of these together, cytokines and adhesion molecules, basically help recruit, activate, stimulate, and warn other cells of our immune system. So how does our immune system see these foreign, how does our immune system even know what a bacteria is? Because remember, we're born with this. Believe it or not, we are born with the ability to recognize bacteria as foreign. And that knowledge was given to us by our mother and father. And again, if we want to go way back in time, it was given to us. We had this ability before we were even really considered human. Our ancient ancestors had these genetically encoded ability to recognize foreign invaders, and they're called pattern recognition receptors. Because remember, this is an ancient war, this battle between multicellular eukaryotic organisms and the bacteria that want to invade them and eat them has been going on for as long as these two organisms have existed. So we're talking a couple billion years here, or at least 1.5 billion years of this war. And so we have these very specific receptors on our cells and in our cells called pattern recognition receptors, PRR. And one there's different categories of pattern recognition receptors. The one that I want to talk to you about are pathogen-associated molecular patterns. So they're called PAMPs, P-A-M-P. There's also, I think there's M-A-M-P, MAMPs, and also DAMPs, D-A-M-P, which is, I think, damage-associated molecular patterns. So we have these receptors that can recognize pathogens. We have them that can recognize damage, and so on. Because remember, our immune system has to take care of damaged cells too. Like when we have a crushing injury to our body, or we have a sprain where we have some necrosis occurring, or a anoxic situation in our body, all of those cells that are dead, our immune system has to clean that up. If they just sit there, they're going to rot and bacteria are going to invade, and that's a problem. So we need to get rid of those damaged cells and tissue as well. But let's focus on pathogens for now. So pathogens are recognized by these pathogen-associated molecular patterns. So they're in three places. So they can be on the surface of our cell to recognize external pathogens, right? So bacteria that are in the surrounding media or the surrounding liquid. We can have these PRRs, these pattern recognition receptors, on the inside of phagosomes and endosomes. So imagine if this is a phagocyte, if this cell is a phagocyte, it's going to engulf bacteria. And before it kills the bacteria, before it dissolves it in acid with lysozyme, it wants to recognize that bacteria, and it's going to recognize it using that pattern recognition receptor. And then finally, in all cells of our body, we have pattern recognition receptors that are just kind of floating around the cytoplasm of our cells. And they're going to recognize damage inside of our cells. And as soon as they recognize that damage, they're going to tell that cell to kill itself and basically commit to an apoptotic pathway so that cell can't cause further damage. So there's three different pathways that these PAMPs, there's three different pathways that these pattern recognition receptors can take. Again, external, inside a phagosome, or internal. So only two of these have to do with PAMPs, though. Which of these two, if you had to guess which two are associated with pathogens, which would you guess? Well, this one and this one, right? Because we're not going to have bacteria inside our cell necessarily, but we will have bacteria. We definitely find bacteria in our external environment. And we would definitely, if we're a phagocyte, we're definitely going to find bacteria engulfed in a phagosome. So what do these patterns, again, we're calling them pattern recognition receptors. So what is the pattern? What are they recognizing? Well, they're recognizing hallmark characteristics of bacteria. Or I shouldn't say bacteria, of microbes. For example, peptidoglycan. What do we know about peptidoglycan? Only, ever, and always in bacteria. Flagella. Who has flagella? Bacteria. LPS. Where is LPS? It's on the external cell wall of gram-negative bacteria. Lipoteichoic acids. Those are also, we didn't talk about them too much, but they're also components of the cell wall of bacteria. Where do we see double-stranded RNA? There's only really one place that we will ever see double-stranded RNA, and that is in a virus. Remember that some viruses are RNA viruses, and they're double-stranded. In fact, I want to say the coronavirus is a double-stranded RNA virus. So if a bacteria sees if one of our cells sees double-stranded RNA, it's immediate. No, you don't belong here. You're not in the club. You're not one of us. You're not self. I recognize your kind and you don't belong here. It's recognizing a pattern. If it sees peptidoglycan, it knows that bacteria are present, and it's going to alert everybody because it's recognizing a pattern. Again, it's generic, right? And it's very fast acting. Now, there are certain categories of pathogen recognition receptors. For example, there are toll-like receptors, nod-like receptors, and rig-like receptors. I think there's a couple more categories too. Let's not get bogged down in the details. Let's only talk about toll-like receptors. So they're called toll-like receptors because I think the guy that originally studied them and characterized them was a German scientist. His last name was Toll. So we call them toll-like receptors. And this is a category of receptors that are found in macrophages and dendritic cells and some other cells of our immune system. And again, remember, these are very common in most multicellular eukaryotic organisms. I think that's just so cool that we share these pattern recognition receptors with our kind of cousins that are like way far removed from us. So where are these toll-like receptors located? Remember, in a phagocytic cell, they're going to either be directed to the outside of the cell, or they're going to be in a phagosome or an endosome when they engulf that foreign pathogen so that they can recognize them. There's lots of different toll-like receptors. There's at least a dozen of them, and they bind to different types of pathogens, and this leads to activation of the cell that it's attached to, the macrophage or the dendritic cell or whatever it might be. And then this cell, this macrophage or this dendritic cell, is going to start secreting cytokines to warn everyone else that they have found a pattern that suggests a pathogen is in the body. So normally when I put this slide up in class, like if we're in a normal class, I tell the students that they have to memorize this for the test, but that doesn't work, and I don't want to stress you out, so I'm not going to use that joke on you. It's so funny. I know, I'm hilarious. But I just wanted to put this slide up because it shows, and believe it or not, this is a simplified schematic. This is a very, very simple view of how toll-like receptors can work. So let's kind of break this down and see if we can kind of understand what the heck is happening in this flowchart. So we have a eukaryotic cell. We know it's eukaryotic because we have a nucleus down here. And we have our external environment out here, and we can also see an endosome here. So notice that our toll-like receptors are in both places. They're on our cell membrane. Remember, we don't have a cell wall, and they're pointed outward. And you can see that the different toll-like receptors will recognize different things, different patterns. For example, toll-like receptor number 5 recognizes flagellin. Toll-like receptor number 11, I don't even know what that is, recognizes uropathogenic bacteria. So some external factor or some epitope on bacteria, it recognizes that pattern that are specific to those kinds of bacteria, and so on. Notice here in our endosomes, we have different toll-like receptors. Toll-like receptor number 9, 7, 8. These tend to be a different set. We again, don't get bogged down in the details, but we have different toll-like receptors for our external environment and our endosomes. These are going to recognize antiviral or different types of virus compounds, double-stranded RNA, sometimes even single-stranded RNA. And we're going to start a signaling cascade. So notice that here at the cell membrane, it's going to activate some protein. In this case, this protein is called IRAK4. And then that's going to cause another protein to become activated. And then ultimately what ends up happening through all of this mess is that signal somehow gets to our nucleus, and it's going to activate specific transcription factors. Again, don't worry about everything that's happening here. What's really happening is we have some type of signal transduction from the external environment to the nucleus. And this seems really complicated because there's so many arrows and circles and squiggles in this picture. But again, think about how a smoke detector works, right? A smoke detector is going to take somehow it's going to detect a molecule of smoke, whatever that might be, I don't even know what that is, in a room, right, through its sensor system, and it has to translate that sensor. It has to translate that molecular activation of smoke to some signal, some type of electronic impulse, and it has to send it to the control panel of the building. That control panel of the building has to send a signal to the sprinkler system. That sprinkler system has to interpret that signal and turn on, right? So that's also complex. And so we have these very complex pathways of amplifying signals that will eventually lead to the expression of certain proteins. What proteins? Well, probably cytokines, right? Probably interleukin-6 to let everybody know to get stimulated and get angry and start doing stuff. It'll attract neutrophils. It'll do all of these kind of pro-inflammatory, pro-stimulatory proteins because it wants everybody to get angry. All right, so when we get angry, what do? What happens when our immune system gets these signal responses? So we have our sentinels. They recognize these patterns. They found some flagella it's gotten past our first line of defense. They found some LPS or some peptidoglycan. And so what actually happens there? Well, we can get activation of complement. We can get phagocytosis. So we can get direct action and direct kind of ingestion of these bacteria by phagocytes. We can get inflammation. And then finally, we can also get an interferon response. So let's talk about each of these and characterize them as well. So remember, I think I mentioned that complement in our first schematic, it was part of the sensor system. And so that's kind of one of the cool things about complement is that it's not only a sensor system, it plays this kind of sentinel function in our bloodstream, but it also serves as an effector function as well. So let's talk about what complement is. Again, crazy complicated field of research. So many researchers study complement because it's a very complex topic, and it's also really important, and it's a highly conserved, evolutionarily conserved process in multicellular organisms. So complement is a set of proteins that were originally identified as complementing, in other words, rounding out our immune system. They circulate in an inactive form in our blood, so they're tiny little proteins, and I think there's actually the video. I have a video that you're going to do a quiz on, and in that video, I think they call them like tiny little hand grenades, and so we have at least several billion. I bet it's even trillions of these proteins that are circulating in our blood, and they're basically like little hand grenades that still have the pin in them. Right? And don't get nervous. Don't get anxiety. It sounds really dangerous, but it's not, I promise. And they're inactive and they're just circulating in our blood. And so these proteins are labeled C1 through C9. I think there's actually a couple more that have been discovered, but so we just kind of name them complement number 1, complement number 2, and so on. And they can be activated in a few different ways, and then they have a few different outcomes. And these activation pathways are really complicated, but I want to talk about the commonality. So let's first talk about how we can activate complement. Oh, hold on a second. I'm late on my categories. Let me pause this really quick and tell. Okay, sorry about that. All right, so let's talk about the classical pathway. The classical pathway actually is another point of overlap with our adaptive immune system that we haven't talked about yet. So antibodies are really the major tool of our adaptive immune system. And when antibodies bind to microbial invaders, the FC portion of that antibody that hangs out, again, you don't know what FC portion is yet, but you'll learn it. You'll learn it next week. It's that part of the antibody, and that portion that's kind of sticking out into the external environment is going to activate complement. Another way is the lectin pathway. So there's this protein called mannose binding lectin, and it binds to different microbial invaders. This can directly activate complement as well. And then there's also another pathway where C3b can actually bind to microbial bacteria and other viruses itself. So it can directly attach to foreign pathogens as well. So there's 3 different pathways that can eventually lead to activation of complement. And this is where they all combine. All 3 will form, all 3 of these will eventually form the formation of a protein called C3 convertase. And C3 convertase splits the complement protein C3 in half. So C3 will split into C3a and C3b. And notice that C3b is, excuse me, that protein that can directly bind to microbial invaders. So at this point, once C3 gets split into C3A and C3B, we can have three things. We have basically three common outcomes of complement activation. Let's talk about the first one, and that's inflammation. Again, we talk about inflammation. I mentioned it already a couple times. It's one of the major effector functions of innate immunity, and complement plays a major role in this. So C3A and another portion of the complement called C5A will induce, basically what happens is it causes your blood vessels to become very permeable. So if you think about your blood vessel as a tube of cells that are closely connected to one another, essentially what happens is those cells don't become as tightly connected. And so they become permeable. And this allows cells to kind of sneak past them, especially macrophages and neutrophils. And a lot of water, I should say, too. And this leads to swelling, inflammation, and a better localized immune response. So the first response is inflammation. So the first outcome, I should say, is inflammation. And I shouldn't say one of the outcomes. So it's not like one happens and then the next. All three could be happening at the same time. Now, on the other hand, we can have C3B directly attaching to the external cell wall, and so I shouldn't say cell wall, cell membrane of bacteria or viruses or whatever this foreign pathogen might be. And when C5 kind of goes and attaches to that cell membrane, it's going to start attracting other portions of complement. You can see there's C6, C7, C8, and then one of the really cool complement proteins, C9, is basically like this huge spike. And when you start to recruit C9, it's going to basically jam itself into the lipid bilayer of the cell membrane, and that's going to cause and then it like a bunch of them get together and form a circle, and that excuse me, and that basically punches a big hole into the side of the cell, and that's going to basically ruin the membrane integrity of the cell, and water is going to get into the cell, all the cellular contents are going to spill out, and that, of course, is going to lead to the death of of the bacteria or whatever cell that might be. So that's called the MAC attack. I think it's called the membrane attack complex. I think they mentioned that in the video, and I know they mention it in the book. It's called the MAC complex, the MAC attack. So I had one of the professors that was in my department in grad school studied complement activation, and he had a t-shirt that had it said MAC attack on it. I just remember that. Okay, so what's our last one? So our last one is opsonization, and so this one, again, a bit tricky to explain in person. It'll help. The video that I have in my lecture will help a lot. Remember that a big way of clearing bacterial infections is through direct phagocytosis. So we have these phagocytes, macrophages, dendritic cells, and so on that like to surround and basically engulf these bacteria. But remember that bacteria are slippery. They have capsules. They've had literally billions of years of evolution to produce a slippery outer covering that will help them evade phagocytosis. So what happens is C3b is going to bind to that bacteria, and it basically acts like a little anchor on that bacterial cell. And now when the phagocyte reaches that bacteria, it has specific receptors on the outside of let's say that this is a macrophage. It has specific receptors on the outside of this macrophage that can attach to C3b. So this bacteria is no longer slippery. It's actually primed for eating. So it's like putting like a big flag on that bacteria for the macrophage to come and grab onto it really easy and eat that bacteria. So again, three pathways that can activate complement. They all converge on formation of C3 convertase, and they have three different outcomes: inflammation, lysis, and opsonization. So let's talk about that opsonization in a little bit more detail. I've mentioned phagocytosis again a bunch of times in this lecture, but what is phagocytosis? Remember that not all cells can do this, only very specific cells, macrophages, dendritic cells, neutrophils, and they're recruited. They're either already there in the case of macrophages. Remember, they were kind of roaming through all the various tissues and portions of our body. Neutrophils are recruited to the site by either cytokines or chemokines, right, for taxis, or adhesion molecules that are going to recruit them to that site. And they have specific receptors on their surface that allow them to grab onto the surface of bacteria. So let's first talk about how they get there. Remember that complement has that C5a and C3a protein that is going to lead to vasodilation and recruitment of these macrophages and neutrophils. So they're going to directly recruit these different types of phagocytes to the site of infection. Once they're there, they're going to recognize that C3b on the portion on the outside of the bacteria, and that C3b is basically neutralizing the capsule, right? Because all bacteria that are able to survive in the wild have a capsule, and C3b is our body's way of counteracting that capsule. It's going to cement itself into the side of that bacteria and act as an anchor for that this macrophage, for example, to engulf and eventually digest that bacteria. So once the macrophage kind of does these little, it's almost like an amoeba. It kind of does this almost like a diapedesis thing where it reaches out with a portion of its cell membrane and engulfs the bacteria. It now has the bacteria in what we're going to call a phagosome. So it's almost like a little endosome, kind of like a tiny little stomach within the macrophage. And that phagosome is going to fuse with a lysosome. Now, lysosome is a little packet that's full of the enzyme lysozyme. Remember, lysozyme is deadly to bacteria because it dissolves peptidoglycan. So when that lysosome binds and fuses with the phagosome, we're now going to call that a phagolysosome. And we've now created kind of this tiny little stomach within the macrophage. And that bacteria doesn't really stand a chance. The phagolysosome is going to destroy the cell wall. That's going to ruin the integrity of the internal bacteria, and that bacteria is dead. And basically, that macrophage is digesting the bacteria, and it gets rid of the contents through exocytosis. So pretty cool stuff. This is how macrophages and other phagocytes clear infections directly by essentially eating the bacteria. Now, I should mention, to be fair, that there are some really interesting bacteria that have taken advantage of this process. And there's some bacteria that have evolved to actually recruit macrophages to the site of infection. They actually want to be engulfed by bacteria because once they get in here, they're immune to the lysozyme. So they actually hang out inside these phagolysosomes and replicate inside these macrophages and then take over the macrophage. So it's a brilliant technique because, one, they're hiding from the rest of the immune system, and they basically have a safe place to replicate and take over this macrophage. So they cause huge problems. I want to say that tuberculosis does something like this. The Mycobacterium tuberculosis does something similar to this. Pretty cool trick that those bacteria do. All right. So our next, I think, is second to last effector function of the innate immune system is inflammation. And so when you get into nursing school or whatever grad school you'll hear about the four major signs of inflammation. They're calor, dolor, tumor, and rubor. They're Latin terms. There's kind of, some people include a fifth one called functiolatia, which basically means loss of function. So these are all Latin terms. Calor, of course, means heat. Rubor means redness. Tumor is swelling, and dolor is pain. So these are the four signs of inflammation, and of course the four signs that you have an active infection. This is initiated by the ligation of those pattern recognition receptors. This can happen, again, most commonly, and the one that we talked about was through the was through microbes getting past our first line of defense. So microbes have somehow gotten past our skin or mucosal membrane. But remember that this can also happen from damage. If you sprain your ankle, for example, you have some type of crushing injury that's going to introduce necrosis, in other words, damaged cells. This can happen also. I mean, when you think about when you sprain your ankle or hurt something in your body, one of the outcomes tends to be inflammation. So that swelling and redness, of course, is due to that vasodilation, activation of complement, recruitment of immune cells that are basically going to war with those pathogens that manage to make it past our first line of defense. But now let's talk about fever. And of course, I should say, I mean, swelling and redness and localized heat can all, of course, lead to pain and loss of function. Whereas fever is more of a global, I mean, you can, of course, when you look at a localized infection, it can certainly be red and swollen in that area. And sometimes it can even be warm to the touch. So you can actually get kind of a localized, very mild increase in temperature. But typically when we talk about fever, we're talking about a global response to infection. And of course, this is caused by a cytokine signal. So these cells, the sentinel cells, the macrophages, the dendritic cells are going to secrete a specific cytokine. I think it's interleukin 2, maybe. But these cytokines, there's several of them, and they're called pyrogens. So pyrogens are specific cytokines that are carried to, I want to say, the hypothalamus, maybe. It's a specific part of your brain that turns on, is basically kind of the temperature gauge for your body. And that's going to signal your body to induce a fever. And fever, I kind of look at fever, it's almost like sleep. Like a lot of people study it, but very few people can really give you a great answer about what it really is. So for a long time, the kind of the standard line is that it makes us an inhospitable host. But as you've learned in the class, bacteria and viruses are way better adapted to temperature fluctuations than we are, right? I mean, if our temperature goes up to 104, we really can't survive very long at that temperature. But bacteria can survive, are more adaptable to fluctuations in temperature than we are. But again, there's some research behind it that says that basically we're creating an inhospitable host, and we're basically creating, if we can even slow that virus down or slow that bacteria down a little bit, we're giving our immune system an opportunity to take over. And then the more likely scenario is that fever acts as a global switch. In other words, an increase in temperature is going to turn on specific enzymes and certain and specific signaling pathways that turn on specific responses within our body. For example, it increases the metabolism of our cells, and that can lead to increased recovery time or a faster recovery time. It also signals our liver to basically sequester iron and zinc. So it holds on to iron and zinc and doesn't allow it to become as bioavailable in case there's bacteria in our bloodstream, for example, and it prevents those bacteria from getting, because remember, bacteria need iron and they also need zinc as a cofactor for many of those metabolic processes. And then finally, I just wanted to give you an example of interferon response. Remember, this is an effector function of our innate immune system as well. So imagine we have an infection with a virus, not too hard to picture right now, and we have some virus that's going to infect a cell. So we have early infection, so the early stages of infection, that virus gets in, and the virus is going to start making all the stuff that the virus does. So it could be single-stranded RNA, double-stranded RNA, it could be unmethylated DNA that's contained in the virus, it could be the nucleocapsid, specific proteins, and so on. Well, our cell has sensor system. Remember, it has those pattern recognition sensors within the cell that's going to recognize those viral markers. And what happens is, as soon as that pattern recognition receptor recognizes the presence of a virus, it's going to turn on a very specific gene called interferon, the interferon gene. Remember, interferons are a type of cytokine. And interferon is going to be secreted. So it's a ligand at this point, right? It's going to be secreted by our infected cell. Now, unfortunately, this cell's a goner, right? This cell was the first to get hit, and he's toast. But what happened is he allowed, so by by producing this interferon protein, the signal, the cytokine, he secreted it outside of his cell before that virus killed him, and yeah, led to the death of the cell. And this interferon is going to go to all of the nearby cells. And the cell received that signal. See it right here? And remember that interferon is a warning signal. And so this cell, this infected cell, was able to warn this nearby cell that there is a virus in the area. And what happens now is that this cell is going to synthesize very specific antiviral proteins. So it's ready. As soon as that virus gets into the cell, all of those antiviral proteins are there and they're ready to go. So this is very specific to the host cell, but notice that it's not specific to the virus. Pretty much any virus can elicit an interferon response. And this is part of the reason why interferon response is considered part of the innate immune system, because again, it's not specific. All right, I think that's the last, yeah, that was the last slide in this lecture. I just want to very briefly go over what we talked about here. Remember, we have our first line of defense, definitely the most important aspect of our innate immune system, that intact, healthy skin and mucosal membranes and those antimicrobial substances that help. Our sentinel cells like macrophages and dendritic cells that have the pattern recognition receptors, in addition to our complement system that's just kind of circulating throughout our blood and waiting to get activated. Once those sentinel cells communicate, they recognize foreign pathogen, they're going to communicate with the rest of the cells of our immune system to have these effector functions: interferon response, phagocytosis, activation of complement, inflammation, and of course part of inflammation is fever. All right, so that does it for lecture number 10. That was a long one. I think I talked too much. I'm sorry, I talked a lot in this one. And I also have some additional videos that will probably be very helpful in helping you kind of understand this really, really complex topic of innate immunity.